parasites. We have all heard the horror stories about these creatures. A parasite is an organism that lives on, in, or with another organism called the host. The parasite relies on the host for survival, whether that is for food, travel, reproduction, or to complete their life cycle. Don't be mistaken though, they do need the host, but they are actively harming the host, which is what makes them a parasite. And there are many types of parasites. But in the spirit of Halloween, we will be talking specifically about zombie parasites. Hey guys, it's Oliver, and welcome back to my YouTube channel, Exploring Exotics. Now, when I say zombie parasites, I am referring to parasites that use their hosts by taking over their body, manipulating them, and turning them into something that we would recognize as a zombie. In these cases, the hosts will be invertebrates, and the parasites will range from worms to fungus to barnacles. So beware, the things that you will learn in today's episode may just be stuff of nightmares. Let's begin with a parasite you have probably heard of. Meet Nematomorpha, or more commonly known as the horsehair worm. To preface, Nematomorpha is a phylum of large flatworms found throughout the world, but are often restricted to locations around bodies of water. There are around 11 species that occur in the United States, and they are split into two classes. You have Nectonematoida, which is comprised of entirely marine-dwelling species, who as larvae parasitize on crustaceans. And then you have the one that we will be focusing on today, Gordiordia. Gordiordia are parasites of terrestrial arthropods and are free-living as adults. Hairworms, like most of the parasites on this list, are extremely understudied, so just keep that in mind going forward. But what we do know about Gordiordia specifically is that they're estimated between 300 and 2,000 species. However, some speculate that there are actually more than 2,000 species. These worms resemble nematodes, which is where they get their scientific name, but keep in mind that they are very different. Their life cycle has 4 stages. You have the egg, the pre-parasitic larva, which hatches from the egg, the parasitic larva that lives and grows within an invertebrate, and 4, a free-living aquatic adult. The eggs are deposited in water in slender strips, usually held together by a gelatinous material. Most species lay their eggs in springtime in shallow bodies of water, such as pools, streams, and swamps. Once the egg has hatched into the larva, it is time to find its paratenic host. The host is usually the larva of an aquatic insect. The larvae have rings of cuticular hooks and terminal stylets that are believed to help them enter the host, and this is where the horsehair worm will stay until the host metamorphosizes into a more terrestrial environment. They will form a cyst around themselves inside of the paratenic host and lay in wait. Eventually, the paratenic host will get eaten by a terrestrial arthropod, such as a grasshopper, a cricket, a mantid, or a cockroach. Once eaten, the horsehair worm will break open into this new host. This host is called the definitive host. In some species and in some cases, the larva of the parasite will actually get eaten alongside vegetation or drank along with water by the definitive host, skipping the paratenic host entirely. Regardless of how the parasite does it, once inside the definitive host, the larva will live inside of its primary body cavity, absorbing nutrients and oxygen through their body walls, and releasing their waste into the host. And as they grow, it becomes time to make the definitive host a zombie. Once sexually mature, the horsehair worm will complete its life cycle. The horsehair worm will hijack the host and make them seek out water. Once a body of water is found, the host will jump in, drowning itself, while the horsehair worm burrows out of its body. Exactly how the horsehair worm is able to do this hijacking is still a mystery and is still pretty heavily debated. However, research done in crickets and grasshoppers infected by the horsehair worm shows that they produce different proteins in their nervous system than non-infected individuals. So this may be a form of molecular mimicry, where the parasite is the one causing these changes in the proteins. These proteins are believed to act as neurotransmitters, or are used as the body's response to gravity, known as geotactic signaling. The horsehair worm will complete the majority of its development in the host, but is usually not detected until its exit. Once the worm does make its way back into the water, the cycle continues. And I know what the question is. What about humans? Where are we on this? There's no need to worry, these worms are not a threat to humans. Now, there have been cases of finding horsehair worms that have passed through the systems of toads, cats, and even humans. However, those were not hosts. This is instead a form of pseudoparasitism. They're just able to pass through the digestive tract and live. Alright, so that was pretty unpleasant to learn about. Now let's go on to the next one. Meet Ophiocordyceps unilaterus, otherwise known just simply as cordyceps, and even more simply known as the zombie ant fungus. This is a parasitic fungus that infects invertebrates, mostly carpenter ants. Its taxonomy is still being studied, with some research suggesting that each species of carpenter ants has its own unique species of this fungus. So we're just going to make it more simple and go over them as a whole, since there's not really differentiating factors yet. Just remember that some details may vary species by species. The zombie ant fungus? The zombie ant fungus is found worldwide in tropical forests, with few reports of them being in temperate ecosystems. They begin life as sticky spores on the bottom of the forest floor, and when a passing carpenter ant comes in contact with it, it becomes infected. The spores will attach themselves to the ant and will begin drilling into the exoskeleton with a combination of pressure and enzymes. Once inside the host, the spores will begin to feed on the insides. As it feeds, it will spread throughout the ant's entire body. Here the fungus will grow, and about 16 to 24 days after the infection, the fungus reaches a critical stage. This is when the ant's behavior will begin to change. The ant is induced to leave the nest at different times than usual, and it will fail to walk along established ant trails and instead walks erratically. Infected ants are also usually unresponsive to external stimuli, including other ants. Eventually, the ant will experience sporadic convulsions, making it fall from the safety of the trees. There on the forest floor, the ant's movements become aimless. At this point, the ant will no longer be returning to the nest. Instead, the ant will seek out a more humid environment suitable for fungus growth. It wanders for a time before the fungus compels the ant to climb up a nearby twig or low-growing plant. Once situated, the ant will permanently latch its jaws into a leaf vein and lock into it. This is referred to as a death grip. The death of the host follows shortly thereafter. The host will continue to eat the ant's insides several days after death. And once it's ready, the fungus will push itself out towards the base of the ant's head. It will then release spores, and the cycle will continue. The exact biological mechanism that is used to control the ant's behavior is still not understood, but studies have suggested that the fungus actually aims for the ant's muscles and not the brain. In fact, they found out that when the fungus attacks, it usually attacks throughout the ant's entire body, except for the brain. The fungus actually seems to grow around the brain, keeping it intact. The fungus secretes a number of neuromodulatory agents, particularly guanobutyric acid and sphingosine, both of which are released near the central and the peripheral nervous system of the host. The fungus also contains some unusual bacteria-like toxins. These may be impairing chemical signals that allow ants to communicate. Other research suggests that the organism activates and suppresses certain ant genes throughout the infection to alter and control its behavior. Fungal cells are also known to invade the muscle fibers of the host, which may affect the expression of certain genes, which can explain the tremors and the biting behavior, which is all pretty wild for an organism with no brain or nervous system of its own. And on top of all of that, this deadly relationship has seemed to be going on for a while, with a 48-million-year-old fossil of an ant in the death grip of Ophiocordyceps discovered in Germany. Hey guys, it's Editing Oliver. I just wanted to pop in and make a correction. The fossil was not of the actual ant in the death grip, but instead was the leaf with the markings of the death grip. I don't really know how that slips through the script, but I just wanted to pop in and make that correction. Alright, back to the video. And if that isn't weird enough, this parasitic fungus also has its own parasitic fungus. These are referred to as hyperparasites, and they are a thing that we will talk about in another video. And of course the question, can they infect humans all Last of Us style? The answer is relievingly no. The fungus transitioning to a human host from an ant host is extremely unlikely, especially when you remember that they are a little bit particular about the type of ant that they actually infect. Not to mention that the internal temperature of mammals is usually much too high for fungus. And according to the CDC, the types of fungus that can internally affect humans is very slim. Most commonly, yeast infections and athlete's foot. So unless you are a carpenter ant, I would say that there's no need to worry. But if you are a carpenter ant, here is a tidbit for you. Carpenter ants that are found in the same locations as this fungus have a few adaptations that they've acquired. For example, the ants found in these areas are typically more arboreal than the ants found in other areas where the fungus does not occur, which of course limits their exposure to the spores. They also tend to practice social grooming, removing spores from each other before infection is able to occur. They have even been seen carrying off infected members away from the nest and establish ant trails to avoid spreading, and they are known to actively avoid the graveyards of the infected dead. And without further ado, let's move on to number 3. Hymenopepimyces argiographica. The genus Hymenopepimyces contains 8 described species of parasitoid wasp. These range across tropical Mexico and Cuba throughout southern Brazil. However, specifically, Hymenopepimyces argiographica is only found in Costa Rica, and that is the one we will be focusing on today. The ideal habitat of this wasp include good food sources like sugar and nectar, mating sites like some shrubbery, and of course, a good population of their host spider. These spots are usually in some pretty heavy vegetation. Now, the adult of this species is not a parasite itself, but their young are. Once an egg is ready, it is time for the adult female wasp to find the host. Once locating a good host, usually L. argyra, female wasp will paralyze the spider and then lay the egg in its abdomen. This can be done in 2 ways. One, method of attack, where the wasp will simply hover over the host until there is an opening. The wasp will swing down and pin the spider. It will then jab into the host's abdomen repeatedly with its ovipositor, while also injecting the host with venom. The second method is the deceiving method, where the wasp will simply lay in the middle of the web motionless, to trick the spider into thinking that it's a good meal. Once the spider wanders over, the wasp will grab onto them, stinging repeatedly. Again, with its ovipositor. They do this until the spider stops moving. It then injects its ovipositor. Whichever way the wasp chooses, the end goal is the same, to get the egg into the spider. Once the host is paralyzed, the wasp will have 5 to 10 minutes to inject its egg. First, the wasp will use its ovipositor to probe the spider for any existing eggs or larvae. If she finds any, she will actually remove them and replace them with her own. The wasp's egg is pretty much glued to the abdomen, and 2 to 3 days later, the larva will hatch, but will only partially emerge from the egg. This larva will create a large hole in the spider's abdomen in order to feed on hemolymph or spider blood. This makes a hard saddle. The host will continue to act normally for the next 1 to 2 weeks. Then the second instar will emerge. Then the larva will insert a hook-like structure into the hemolymph saddle, fully emerging from the egg. And here's where the scary part really begins. When this larva is ready to pupate to become an adult, it releases a chemical that causes the host to spin a very special web, making a cocoon for the larva that will withstand a lot of different rains or winds. It will consist of several heavily reinforced radial lines that are attached directly into the ground. After spinning this web, the spider will then sit motionless in the middle. Even if the larva is removed before the web building process, the spider will still make the web. After this, the parasitic larva will kill the host with toxins. It then eats the spider and makes a cocoon in the center of the web. Roughly 1 week later, the adult will emerge. And just like the others, we don't know the full details of how this happens, and unlike the others, we actually have no idea. I wasn't able to find any information on how exactly the wasp accomplishes this takeover. I'm sorry. Alright, up next at number 4 is probably one of the most disturbing ones. Meet Sacculina carcini, more horrifyingly known as the castrator barnacle. Sacculina is a genus of barnacles. These are monoxenic parasites of crabs, specifically the green crab. However, they have been found to infect the harbor crab, the velvet crab, and the flying crab. They also belong to a group called Rhizocephala. The adults of the species bear absolutely no resemblance to the barnacles that covered like ships and stuff, but they are recognized as barnacles because of the larva. The parasite's range is the same as the green crab. Usually on coasts of Western Europe and North America, the green crab has been expanding its range to become more established in both North and South America, Southern Africa, and Australia. So it is possible that the parasite also is in those locations by stowing away. However, it is unlikely and we haven't found evidence of them being there yet. These of course live in marine environments, and during their larval stage they are free swimming. The female larva will settle on a part of the crab where its exoskeleton is a little bit more vulnerable, usually on the membrane that is at the base of one of the crab's hair called a setae. She then molts into a form called a kentragon and injects her soft body into the crab. After this invasion, the crab is unable to perform molting. The natural ability of regrowing claws for the crab is also now gone. So far, these are pretty normal symptoms of parasites, so why are these on the list? Well, once inside, the parasite will form a root system throughout the crab, and will castrate the crab if it is male, interfering with the crab's hormonal balance. This will sterilize the crab, and if it is a male crab, will actually change its appearance by widening and flattening its abdomen and making it appear as a female crab. From here, it makes its way through the crab and will push out a sac on the crab's abdomen. Usually, this is where the crab keeps its own eggs, but the sac from the parasite is known as an externa. The part remaining inside is called the interna, and the interna develops tendrils. These are what spread throughout the crab. This takes over the stomach and intestines to absorb nutrients, and they take over the crab's nervous system to allow control. So not only does the male crab now look like a female crab, but it also behaves like a female crab, going as far as performing mating dances. But why would a parasite need to do this? Well, it aids in reproduction and the life cycle of the parasite. The eggs of the parasite are inside of the externa, which are fertilized by male larvae that enter the sac through a pore. The males are tiny, short-lived, and will never become adults. Their only purpose in life is to fertilize the eggs. Hundreds of these eggs are produced daily and will stay in the sac for around 6 weeks. And that is where the host comes in, basically becoming a babysitter. The crabs will actually begin to groom and aerate the eggs as if it were their own brood, and they will do this until these eggs are ready to hatch. The natural hatching process from a normal crab with normal crab eggs consists of a female finding a high rock and grooming the brood pouch on its abdomen. It will release the fertilized eggs in water through a bobbing motion. The female crab will stir the water with her claw to aid the flow of water. The crab will perform a similar process for the parasite eggs. This process repeats with the adult parasite continuing to live in the crab for the rest of its natural life. If the parasite is removed from the crab, female crabs tend to regenerate some of their ovarian tissue, with male crabs usually developing partial or complete ovaries instead of regaining their testes. Sorry if that one was a little bit too much for you guys, but let's go ahead on to our fifth and final one. Meet Leuconchloridium paradoxum, or the green-banded broodsac. The green-banded broodsac is a type of flatworm. It is found in Europe, North America, and Japan. They are most common in moist areas such as marshes, where their intermediate host, the amber snail, lives. It oddly starts out in the cloaca of an infected bird. This is where the adult parasite will feed on passing digested material. They are usually found in birds such as crows, jays, finches, and sparrows. They reproduce asexually and release their eggs into the bird's feces. Some of the contaminated feces will be eaten by amber snails. The eggs ingested by the snail will hatch into myristidia. These will develop in the snail's hepatopancreas into the next stage called a sporocyst. The mature sporocyst consists of a number of branches spreading throughout the hemocell. Some of the branches develop into long tubes ending in a swollen broodsac. Normally only 2 or 3 are mature simultaneously. This is where one or both of the snail's eye stalks become occupied by a mature broodsac, which completely transforms their appearance. The eye stalk becomes swollen, colorful, and pulsating at a frequency up to 80 pulses per minute. The host snail can no longer retreat its eye stalks at this point. The brood sacs inside of the snail are also incredibly visible and mimic a caterpillar, which encourages the consumption of its permanent host birds. This is a textbook example of aggressive mimicry. The snail will also manipulate the snail's behavior to make them, again, more appealing and sought out by birds. In one study of amber snail hosts, the infected snail stayed in better lit places for longer and sat on higher vegetation and were even more mobile, whereas 53% of infected snails remained fully exposed for 45 minutes of the observation period, while the figure was only 28% for the controls, or the non-infected. We aren't sure how these parasites do this, of course, but hopefully we will be getting more answers as these creatures are more prominent in the news than ever. As long as we stay curious, I am confident that one day, at least, we will find out how these zombie parasites control their hosts. And in the meantime, we can hope it doesn't happen to us. And I'm sure as some of your stomachs are relieved to hear, that is all I have for today's video. Comment down below which one you thought was the scariest. Check out the description, all of my links, including my Instagram account, my art Instagram account, my Facebook group, and my wife and I's Etsy, and most important, all of my links for today's video will be there. So if you are curious about any of this information, go check those out. And of course, subscribe to this channel if you are into this kind of content, or really any animal kind of content. I upload every single Sunday at 10 a.m. Eastern Standard Time, alternating between exotic pet education and exotic pet care, but you can hit the bell if you don't want to remember all of that. And as always, I will see you guys next week. Have a happy Halloween. Bye.